Boy. You know, I'm looking at the comments during those 2 minutes of the intro song playing, and people are getting set for a feisty one tonight. Ignore the trolls. Everything's going to be okay. Bite your lip. Hang on tight. Listen, I'm convinced that one guy started this whole thing, and he's been addressed. I spoke to him today, and I told him, dude, stop acting woke right. What do I mean when I say woke right? Here's what I mean. Somebody who is absolutely convinced that everything, no matter what it is a conspiracy, and that you have been fooled and duped, and everybody, people like me, people like Wayne Peters, people like Clyde Do Something, people like Jasmine Lane, people who do independent media are all part of it. We're all grifters. We're all being paid by the Conservative Party or the Liberal Party or the government of Canada or some unknown entity to spread falsehoods. 2 + 2 = 5, just on the other side of the political spectrum. Some of those people, I believe, we can still bring back. Some of those people are gone forever, and many of them are simply AI trolls. Ignore them. They're designed, and they are designed specifically to cause division within us. All right, so hang on tight. This is going to be a good show. Tonight, once and for all, we will dispel all of those myths and all of that disinformation about Universal Ostrich Farm. Katie is going to join us tonight, and Dr. Stephen Pelech. He is a UBC researcher. He's ready to go as well. So all of that begins in just a couple of minutes here on the Shadow at Night live stream, which is brought to you, as always, by CanadaNanoSilver.com.
Comparing Crest toothpaste to SilverCeuticals toothpaste. First, we're starting out with Crest. It's a classic. It's an original. It's what I've used most of my life. But that being said, it does have its problems. First off, the consistency is so weird and thick, and I get it's a paste, but it really doesn't spread that well. And I must also say that I'm not in love with the taste. I get toothpaste isn't supposed to taste good, but why shouldn't it? But that being said, it does get the job done. Next, we have SilverCeuticals. They have 3 flavors, but today we're going with Bubbleberry. And already it smells so good. So I'm excited to see what this tastes like. The consistency is also a lot smoother, and I must say it spreads way nicer in your mouth.
This literally tastes like fruit bubblegum.
Yeah, this toothpaste was pretty good. I actually went in for seconds, and it's infused with nano silver, so it helps remineralize your teeth and keep them fresh throughout the day.
100% go with SilverCeuticals.
And here's the results. What's your favorite toothpaste?
CanadaNanoSilver.com. Oh my God, nanoparticles. They're designed to turn you into a robot. It's bio-digital convergence. 204-808-9292, CanadaNanoSilver.com. Some excellent products. They work incredibly well. Check them all out on the website and then go ahead and start buying. Hello, Kim. How are you doing tonight? Kim's in Oka River watching on Facebook. Hello, Nikki and Rita. Kev, shop for the best toothpaste, he says. He's Canada Nano Silver, obviously. Kevin and Anna. Hello, Michael. Beep, bop, boop. Phil is watching from Wainwright, Alberta tonight. Hello, Phil. Welcome to the show. Nell, Kevin, Mike, Lori, Gloria, Colleen, all watching on Facebook tonight. Lots of people watching on YouTube. Lots of people watching on Rumble, like You Go Girl. Patricia 0711, Lynn's 321, Amnesty Anarchy. Ooh, woke right? I don't think so. Yapasco, Lynn's 321, all those great people watching on Rumble. Guys, don't forget to share this around. Like and share. Get this out to as many people as you possibly can, okay? Thank you very much in advance. Katie Pasitney from Universal Ostrich Farm and Dr. Stephen Pelech join us in 30 seconds. All right, welcome back. Here's our guests. There's Katie from Universal Ostrich Farm and Dr. Stephen Pelech. He is a University of British Columbia professor. Welcome to the show, you two, and thanks for joining tonight.
Thank you.
Okay, first thing I want to do, by the way, Katie, how are things at the farm? Is everything okay?
Doing good. 154 days, happy, healthy, natural immune animals. Yeah.
Okay, excellent, excellent. So listen, I showed you that video yesterday, the one that's been making the round. So I want to play it for everybody in case people haven't seen it. This is an out of context clip from left-wing corporate media from 3 years ago. This is before Katie knew all of the stuff that she knows today, and may have used some wrong wording, let's just say. But anyway, she'll comment on it once we've seen it. It's about 50 seconds. Here we go.
Then there's been an antibody program, which is the latest and really amazing thing that the farm's been working on. And actually half of this farm is injected with dead COVID-19 vaccine and the COVID variant. And they've been working on building antibodies because these birds just build such amazing, strong antibodies. So they actually don't have to harm the animals at all extracting the antibodies. And they use the ostrich eggs to get the IGUIs and what is needed to do some medical research and science to hopefully change the landscape and about therapeutics. And we can neutralize, we've actually have proof now that we can actually neutralize COVID-19.
Okay, so let's talk about that video. That's the one that the woke right has been using to vilify the farm the last couple of days. What exactly did you mean when you said dead virus, COVID-19 dead virus, Katie?
Well, that long ago, when we entered this journey, I definitely didn't understand it like I know today. And I definitely didn't have the experience that I've had with such an amazing doctor like Dr. Stephen Pelech. Steve, you've been instrumental in teaching us so much, and I appreciate that. So back then, my terminology was very rough around the edges. I've learned that correct me if I'm wrong again, please, Steve, but it's one simple protein, and it's an antigen, not a vaccination. So one simple protein is then it out of a whole genome sequence is then what would you call it, Steve? It's not simulated, but it's
it's the actual protein from the virus.
Because I want this to be very clear, yeah.
But it's artificially produced.
Yeah.
So normally the virus, its genome encodes 28 different proteins, and one of those proteins is the spike protein. It's the same protein that you'd find in the COVID-19 genetic vaccines that's produced by your own body cells. But in this case, what happened was they're a collaborator they were using what we call lentivirus constructs. So these are viruses that are engineered to contain the gene from the virus so that when they infect cells in culture, I have to emphasize, not in the ostriches, but in culture, to produce the protein, the spike protein. That protein is purified, and then the protein is injected into the ostriches, and they develop an antibody response against the spike protein, which will protect them against the virus itself, which has the spike protein on the surface of the virus.
It's always good to have a multi-level expert on the show to make sure that we get things right here. Dr. Stephen Pelech is a researcher. He's a professor at the UBC. And I know you've done this a million times, Steve, and you probably get sick of it. But can you give us the credentials that you've got, what you do? And I know that you had a pretty rough go from some of the woke left during the COVID-19 pandemic as well.
Yeah. Well, I mean, I'm on faculty at the University of BC for the last 38 years as a full professor in the Department of Medicine. My research is very basic research, usually into the cellular signaling systems that operate in living cells. But I am also one of the co-founders of the Biomedical Research Center, which until just recently, when it was decommissioned, was an immunology institute at UBC. And I've published in immunology journals, and I've published in viral journals. And the work that I've done has also been with my company. I'm the founder of two companies, most recently Connexus Bioinformatics. And Connexus actually did a 4,500 person study to look at the levels of antibodies produced in people in response to natural infection and also from COVID-19 vaccines. And then more recently, we've been looking at the immune response to the influenza virus. So we have some data actually from the ostriches with their eggs, as Katie's mentioned in the video, of the IgY antibodies. And I'm also one of the founders and the vice president of the Canadian Citizens Care Alliance, formerly the Canadian COVID Care Alliance. So I'm basically co-chair with Dr. Chris Shaw of the Scientific and Medical Advisory Committee. So those are some of my credentials.
You know, interestingly, I know you wrote an open letter during the pandemic response, probably about 3 years ago. And all I could find were the criticisms of that open letter. Still, today, I could not actually find the open letter to read it this afternoon. Believe it or not, I went like 27 pages deep into Google to find your letter. And I even went on to other search platforms to see if I could find it. Nope. It looks like it's been scrubbed.
Well, exactly. And this is one of the reasons why, for example, we at Canadian Citizens Care Alliance decided to start producing books, because it's harder to scrub those.
Right.
So we have 2 books. One, Down the COVID-19 Rabbit Hole, a pandemic of basically ignorance, fear, and greed, is the second book, COVID-19 Pandemonium. But the first book actually deals with the science of COVID-19, and the second book deals more with the politics of COVID-19. So those are kind of hard to scrub.
Can we talk about, why people may be so up in arms about what is going on at the farm? Because it's so easy to conflate what, the big pharma companies did, especially in the past 5 years, with what actual researchers working with natural things like ostriches, such a big difference between the two, but yet people conflate them instantly, and they will immediately start pointing fingers and say, well, you're producing radioactive glow-in-the-dark masks.
Yes.
and crazy stuff like that, right? So, we've got to be able to make that difference between what actual researchers like yourself do and what the money, as you pointed out, what the money does. It drives production and moves things along too quickly, I'm certain, without proper testing. No guardrails in place along the way for many of these things that are coming out, and not just for COVID, but for many things. Yeah.
Yes, I mean, this is the problem. I mean, I think the situation with the United Ostrich Farm is a little bit different in terms of the commercial interests were really driving more the COVID-19 pandemic responses, and the government was doing its best, it thought, to try to protect the public. I think that's what's probably happening with the Canadian Food Inspection Agency, but they're very bureaucratic, and they've been following guidelines that are not actually keeping up with the scientific data as it unfolds with respect to the H5N1 pandemic. And so they are overreacting on the side of caution, but that all comes with great costs, that are associated with this. This is what we saw what happened with the COVID-19 pandemic. We actually saw the harms that were generated from the response probably had a greater negative impact than the virus itself actually had. And we're seeing the same thing repeated now with the with the influenza H5N1 virus and the United Ostrich Farm.
Well, I hope it doesn't come to that. Katie, can you explain the process of what actually happens when you inject an ostrich there? You started telling me this before I left, but I was so concerned about not forgetting anything that I didn't get it all. What exactly happens, and what is the process from beginning to end? Because you're working with a laboratory in Japan.
Yeah.
So what is, from beginning to end, what exactly happens? What are you responsible for in terms of research there at the farm?
So we administer the antigen, not the vaccine. Let's be very clear, the antigen. And in 4 weeks, so they start to develop an autoimmune response to whatever they've been given for an antigen. They create antibodies, very robust, strong antibodies, because they are a prehistoric bird. The same amount of an antigen that is given to a chicken is given to our 350-pound hen. But they still can create the most robust, amazing antibodies. I believe if it's scientific, my knowledge is right, one ostrich egg is equivalent to 100 chicken eggs worth of antibodies. So they're very robust, strong. The antigen is only given to our hens. And in 4 weeks from that antigen being given, the antibodies get put down into the egg. Then you can take the IgYs, extract them from the egg yolk. And then from there you can you have to purify them. I don't even know like what is done in the lab fully. I'm still learning that myself. So I'm going to leave that to Dr. Pelech. And there you can now start to create therapeutics and antibody therapeutics that can help animal and human health naturally. And I think that's what we're really trying to defend here is, everybody gave me a really hard time in that video. I want to give a very small backstory to that video. It was probably about 35 degrees while I was doing that video, trying to think about all these words as well, getting pecked in the back of the head, pecked on the arms. And I was just learning all of that, but we were also under threat of fires. So we came down and hunkered down at home because we were on evacuation order, which we said we were not leaving because we weren't going to leave the animals then. And we are not leaving the animals now with this threat. So I'm actually very proud of myself for handling that moment the way I did. And yeah, I used the wrong terminology, and it's really sad that in the world that we're in right now, with the threat of war and everything going on around the world, and viruses and the threat of the pandemic, that we are not more. We're being so divided, and I don't understand how we're not coming together and becoming united more at a time like this, because there's one thing that I think we can all agree on, and that is that this has to stop. All these animals that are unnecessarily getting killed without being properly tested due to bureaucratic overreach, they don't deserve to die. And I think we can all agree upon that. So that was my backstory. Sorry, I had to. But
that's all right.
So but the process then, these therapeutics can help animal and human welfare. And this is something so extraordinarily beautiful, and we should all be really supporting it. And chicken antibodies have been used for how long, Dr. Pelech?
Oh, chickens, I remember when we first started our institute back at UBC, that was over 30 years ago. Well, yeah. 38 years ago, and we were some of the investigators were using chickens to produce antibodies that they were using in their research.
What is it about birds? What is it about specifically about ostriches that produces such a response? Why are they so good for this kind of research?
Well, actually, the ostriches' eggs themselves, the yolk is where the IgY antibody is concentrated.
What is the IgY body? Sorry, you were about to say that.
IgY antibodies are very similar to the kind of antibodies that we produce in mammals that are in the bloodstream. These are IgG antibodies. When you have a response to antibodies that are in an infection from, let's say, a respiratory virus like RSV or influenza or COVID-19's SARS-CoV-2 virus, they are actually IgE, but primarily IgA and IgM antibody responses. So these antibodies are very, very similar to each other, but they can form complexes easier with the IgM and the IgA antibodies. So they give you much better protection. But in the bloodstream, the IgG antibodies, they last up to about 21 days, whereas the IgM and the IgA antibodies, you're looking about 4 or 5 days before they have to be replaced by new antibodies. So the IgG antibodies that are in mammals, the equivalent is the IgY antibodies that are actually in the ostriches. So these IgY antibodies, however, are extremely stable to heat. You can almost boil the antibodies and they still work. And you can subject them to very acidic conditions or very alkaline basic conditions, and they still work. So they're actually much more durable than the kind of antibodies that we get from rabbits, for example, which is where in my company, we produced about 1,600 antibodies against cancer proteins for research purposes. So I have a lot of experience in producing antibodies and purifying them and characterizing them. And so the IgY antibodies, they're actually much better for industrial applications, or if you need a lot of a particular antibody in large amounts, and that would make it suitable for developing, let's say, diagnostic tests or therapeutics that you could inject into people, that they would then fight the virus with the antibodies that you're giving to, in this case, the patient. We were treating COVID patients, for example, with monoclonal antibodies, but you could produce these kind of antibodies in the ostriches extremely quickly. And they're what we call polyclonal. So an antibody recognizes a very tiny portion of its target protein, the antigen. It recognizes what we call an epitope. And so when you have a natural response, as you would get from these ostriches from injecting the protein, you're actually making a whole series of different antibodies that target different parts of the same protein and is much more effective at actually neutralizing that protein. So this is where some of the advantages are of the kind of process. And then these ostriches themselves, I think, Katie, we're looking at they can lay, 30 to or more eggs in a season. And each egg contains the material that would, for me, would be the equivalent to about 10 rabbits.
They're very big.
They're very big. And
Yeah.
it's a very durable, and they can be easily stored. And antibodies themselves, even the regular antibodies that we've produced in rabbits, rabbit polyclonal antibodies that are mainly IgG antibodies, we can keep them in refrigerators for even over 10 years and freezers for over 20 years. So this is a very durable product that, in fact, can be used because of their stability very, very easily in industrial applications.
Is it because, Stephen, is it because these birds have been around on the planet for so long and they've developed this over such a long period of time?
Yeah, I think for most of the animals that are on the planet, like 99% of all animals have gone extinct on the planet. Those that we have today, they've evolved to have very sophisticated immune systems that offer different types of protection. We have, adaptive immune systems, which basically get educated when we are exposed to a virus or some pathogen. We eventually develop antibodies and it clears the pathogen. But the next time we encounter that pathogen, we don't get sick, or if we do, it's very, very mild. We also have what we call, you have the adaptive immune system, and we have an innate immune system. And this immune system is non-specific. It's your first line of defense against the first time that you may be infected with a new virus or something. And it provides a fairly high degree of protection. It's very high levels of this activity in children. So that's why they actually fare quite well when they're infected for the first time. But as we get older and our adaptive immune system becomes educated in what's out there and much more effective, we don't need as high an innate immune system. But it does require that we constantly get exposed to these viruses, bacteria, in the environment on a regular basis as we age, just to tune up our immune system. If we don't do that, then we start to lose our adaptive immune ability. So
living in a bubble is not a good thing.
Not a good thing. No.
And would you say you're like living in a bubble when, by eradicating all of these animals that have been here for decades, and we're losing our genetic diversity, we're losing their immune systems that have been exposed to so many natural elements that we probably don't even know how many for their antibodies. We eradicate all of those, and their answer is for us to repopulate with a whole bunch of young animals, which I'm sure Dr. Pelech will tell you is not a good idea, because now you have a bunch of naive, weakened immune system animals that need lots of time to develop natural immunity and a strength in their immunity. And then they're bringing up the mutation, that this virus has the ability to mutate. Well, viruses mutate. That's their job. They're always trying to develop a different way to get stronger. But the weaker the animal, being younger and needing more time to develop that, they're more susceptible to the mutation. Are they not, Dr. Frey, I think? Are they? Sorry. I think it's really important because they want people to repopulate with young animals.
That's right. So young animals, they're going to be naive. That means that their immune systems have not been exposed to the virus.
Yeah.
And so if you get an infection of a naive population, the virus spreads very quickly through that population and outside of it. When you have a sufficient number of the animals in the population or humans that have got natural immunity or even vaccine-induced immunity, then you have what's called herd immunity, and it's much harder for that virus to propagate.
Is there anything that could go wrong with what they're doing at the farm in terms of danger to humans?
No, it's pretty minimal, especially now. I mean, the reality is that the H5N1 virus is an avian virus. It's actually evolved to be able to infect bird cells. The surface of bird cells have what we call a sialic acid. That's what the virus latches onto so that it can get inside the cell. The sialic acids that we have in mammals is different from the sialic acid that the virus is binding to in birds. So we have influenza viruses that have evolved to be very effective at infecting birds.
No, hold on, Dr. Pelech. They're suggesting this is a brand new, never before seen H5N1 in Canada that can jump to humans. Now, there was one person, I think Katie had her on. I was just about to ask you if that's nonsense. One person who was infected with this somehow, some way, right, Katie? One.
Yeah, they say there was one confirmed case in Canada, and that was Jocelyn, who visited the farm.
Yes.
And she was the one who said she watched it because they started, the news and the media was using her story for the reason for killing all the millions of animals. And she sat back and watched that and said, how dare they use my one story to give them the go-ahead, the green light to kill millions of animals, most that have not had testing. So we're killing the survivors. And so she came out to the farm. And she said, I want to go and I want to support the ostriches. And we had her out here, which was a beautiful visit. What a beautiful family. To find out that our government is actually taking their blood and banking their blood because they're acknowledging that their family has antibodies. So we're identifying that antibodies might be a good thing for humans and humans, but all of a sudden antibodies don't exist when we come to ostriches.
Interesting.
I talked to Jocelyn's mother actually on the weekend, and I learned a lot more about what the family has gone through. So, in privacy, I don't really want to go into details, but the situation for actually Jocelyn, she was ill with an infection, went to the hospital initially, and was basically sent home with Tylenol, and the situation got worse. I suspect that the H5N1 was probably a secondary infection because the primary infection wasn't treated properly. But she has comorbidities as well, which exacerbate her condition. So if you look at her case, it's the only one in Canada that's for at least 6-7 years that's been documented. We have probably over 70 cases in the United States. There's been one death in the United States of an elderly person who also had comorbidities and chickens in the backyard. So they suspect that's how that individual got infected. But we don't know how it was that Jocelyn got infected because she had no association with any farms or animals that were infected. Now, when you look at the 70 plus cases in the US, most of those cases are actually from dairy farms, not chicken farms. Mm-hmm. And in those cases, what we're seeing is that most of them had no or very mild symptoms. And so this is what happens is this virus that's evolved to infect birds is not very effective at actually being transmitted and propagated in people. So H5N1 virus, these are two of the proteins that distinguish the various strains that are out there. It's been around for over almost 70 years. There's been no cases of pandemics with H5N1 in humans. Mm-hmm. In all the rare cases of where we've seen a human become infected, And usually nowadays it's because they tested with a very sensitive test for the virus, not necessarily that they were sick. What we see is there's not a single documented case of H5N1 being transmitted from a sick person causing sickness in another person of H5N1 so far in any of the records that we have. So this is not the kind of virus that you're going to get a pandemic generated, even with mutations. And with the mutations, we're still looking at the various influenza strains that are out there. They're still about 80% or greater identical to each other. So when you have an antibody response against one influenza, it actually is pretty good at protecting you against other influenza strains. However,
because they're very close, right? And they talked about that during I can't believe how much we learned during the pandemic about viruses. And by the way, quickly for the woke right crowd, I have a question for them, for you, Dr. Pelech. Are viruses real?
Well, I'm not only am I convinced, but we have tens, probably hundreds of thousands of scientists going about their research every day studying viruses. We have the genomes of probably more than 7,000 different viruses. In my case, first of all, you'd never be able to develop a vaccine against the virus if we didn't have the genome of the virus. We wouldn't be able to develop rapid antigen tests. We wouldn't be able to develop drugs that actually effectively treat viruses, they're antivirals, if these didn't exist. We can see them in electron micrographs. So when they're so small, you can't see them with the light microscope. So when you have Koch's postulates that people talk about that were developed, these are like over 180 years ago. So they barely knew that viruses even existed at that time. Since then, Koch's postulates, even Koch himself actually rejected some of his postulates in his lifetime. And really since the 1960s, we have the Hill kind of guidelines for linking a disease to a virus, and we use those guidelines really in research now. But I think a lot of the people are questioning about the existence of viruses. These are the very same people that often question the existence of even proteins and DNA and genetic material, which is even smaller. About 1,000 times smaller. So it's
a good thing we got PCR testing. We can find that stuff now.
Yeah, the only problem is it's the PCR testing it's very sensitive. It can be very accurate if used properly, but it's often used at what we call thermal cycles that are very high, that have a very high rate of false positives. So, for example, in the case of the Universal Ostrich Farm, when they had 2 sick birds that died and they were tested, with swabs, and they came out positives, but these were only for the H5 gene. They weren't for the gene for the N1. So we didn't even know at that point that they were H5N1. And then when they get this information, basically, I guess it was 42 minutes after they got the result?
Before we even got, so they weren't able to test for the N1. That's the Winnipeg lab, the accredited lab. And we only tested it positive for the H5. And 41 minutes later, from those results of the Abbotsford lab, they already signed our call order. Right. So without the full like it going to the accredited lab, our order was signed.
Do you know what cycle rate that was run at, Katie?
Well, actually, no one's given us the actual cycle number. What we do know is it's over 36 cycles.
Yeah.
And we know that at 36 cycles, when you're doing a PCR test, you can expect about a 90% false positive rate.
And then so a bunch of farms in Fraser Valley, starting to look at their lab results, are 39 cycles. I was asking them, there was a farmer, and I said, all of his partridges and chickens were culled. And I said, do you have your lab results? No. Have you been compensated? No. I was like, you don't have your lab results and no compensation, and they've been in and they've killed everything. So I said, please request your lab results and ask them how many cycles they ran your PCR tests. And his was 39.
Yeah, and they had I think one farm that was about the same time as your farm that was infected. Yeah. They killed basically they got their lab results, and within 3 days they had killed 17,000 all of his chickens.
Yeah.
I'm gonna throw a comment up on the screen here, you guys, because no matter what we're saying here, we're talking to an actual immunologist, a scientist, a professor at UBC who's been working in this field for years, decades. And here's somebody saying problem buying into this garbage. Now, if you mean the things that we're talking about right now, then there's absolutely zero chance of you ever recovering from your woke mind virus, your woke right mind virus, JJ. You really need to be able to look at the actual facts of what's going on here. Is there anything nefarious, Dr. Pelech, that could happen? Not necessarily at the farm, but at the lab in Japan. Is there anything that anybody could do to change the sequences of these ostrich eggs and turn this into a new pandemic? Or
no, this is a very hard virus to mutate. There is a couple of mutations that in the literature have identified that can make it more easily transmissible to a mammal, and it clearly has been passing through different types of mammals, including both marine animals, seals and the like, as well as in even dogs have been infected. I mean, this is the thing that I find very fascinating. On the one hand, the Canadian Food Inspection Agency is saying that these ostriches being infected, that their excrement and the virus has been shed and it's contaminating the soils surrounding on that site. And what we do know, of course, with influenza virus is it's a respiratory virus. It's not that easily obtained off the surfaces, what we call fomites, where you could touch something and then it comes easily, you pick it up in your mouth or something and you get infected. This is generally like RSV, like SARS-CoV-2. Influenza virus is breathed in through droplets, tiny droplets that then go into your lungs, and then you can get the virus establishes itself because there's receptors on those cells that allow the virus to attach. Now, the thing is, the one study that was cited in court by the Canadian Food Inspection Agency was where they purposely contaminated water, and then they took that water that was in pond or lake water, and they filtered it with a filter that would capture anything that was above 0.2 microns in size. A micron is a millionth of a meter. So the virus itself is 0.14 microns. So what happens is the virus can go through the filter, but pretty much larger viruses, all bacteria, all fungi, all other cells cannot do that. Then they put that in cold storage in the dark for up to a year, and they found they could still get active virus from that. Now, the thing is, in the wild, sunlight has ultraviolet radiation, and it just mutates any DNAs on the surface of the ground. And anything that's in the soil, microbes completely digest it. So this is, if you think that sounds, and you're worried about that, put this into perspective. If I live in Richmond, BC, we have tens of thousands of geese and ducks that come to our parks, along the dikes, in the schoolyards, laying excrement about every 3 or 4 inches from each other. And no one has issued any panic warning about the influenza H5N1 coming from these birds, where it's just rampant at one point in their population. And we didn't have any large cases, and no cases, except for this one child, of H5N1 in people getting sick.
And where are we going with that, right? That's scary too, because if we don't dispel the fear out of this and we don't educate, then what's going to be next? Are we going to lock down our beaches? And are we going to lock down our lakes? And are we going to be, right?
Wouldn't be the first time, Katie. Wouldn't be the first time.
No,
yeah.
But fortunately, I think in the wild bird population, we're seeing the level of the virus has actually waned, and it's doing exactly what we saw with COVID-19. Initially, you had a virus which was more pathogenic. It caused more people to get sick and even die. But as time went on, it was replaced by other variants that were actually even more infectious. That's how it displaced it.
But
they
less
all-
deadly, right? That's how
and
viruses
less
generally
deadly.
work. Yeah.
Exactly. So what happens is people have minimal symptoms. They still go out. They spread the virus, and that virus is able, the less pathogenic and the more infectious displaces the earlier form of the virus. We're seeing the same thing with the influenza, and actually the data from the United Ostrich farm is one of the evidence that we see this happening in real time. So you can question whether or not when the decision to make the call happened, where that really was H5N1, highly pathogenic avian influenza virus, which means that you have a 75% lethality rate or higher. Now we know that these birds actually is closer to 15 to 16% lethality in those birds. So they would have what would appear to be a lower pathogenicity virus, and this is what really bugs me, is that when we were going through the courts to try to look for the call order to be terminated, we first found out about these tests that were done and asked for more information, like you were suggesting, Shadow. Well, what's the cycle threshold number that they were using, which they still haven't disclosed to this day. But they said, well, this is an unusual virus. There's a reassortment that we haven't seen before. Well, when I saw that, I knew, well, they must have sequenced the genome of the virus, which they actually did. They had their full genome sequenced by January the 10th, but we only found out in the court case about 2.5 months later, just before we were actually going to be, a couple of weeks before we were actually going to court. So when we get that information, then we found out that it was actually not the highly pathogenic H5N1, but it was a mix of low pathogenicity virus with high pathogenicity virus. The genome has 8 separate pieces of chromosomes, or segments, right? So when you get 2 different strains infecting the same cell at the same time in the same animal, when the virus is reproducing and being repackaged to make new virus particles, sometimes you get the mixing of those genes. So this is what happened in this case. And
So it happened naturally.
It happened naturally. Now the question is, did that happen in the ostriches, or did that happen in the birds that were carrying the virus that infected the ostriches? Now, when you have so few ostriches, and very few of them were sick at the time that they took these samples, it's very, very unlikely, because reassortment is a very rare event. You have to have, like I say, the same bird, the same cells, same time, being infected. So this is a rare event. So it's much more likely that it happened in the wild birds, and then this virus propagated and displaced the more pathogenic versions in the wild birds. But what we learned from the court case, where we had basically cross-examination of their experts, we learned that from the Canadian Food Inspection Agency that they have 100 cases of reassortment in Canada. 100.
Not one. Not one at Universal Ostrich.
That's a very rare event, though. I mean, how can there be 100 cases? So something's fishy here.
So what it tells you is that there actually has been extensive reassortment going on in the wild bird population. You now have strains of the influenza virus. You have 2 things happening. One, you have the natural immunity developing in the wild birds because they're not being called if they survive. Plus, you're having the virus evolve so that it's less lethal. So that's the infections. And what's quite interesting is if you look at the situation with the H5N1 outbreaks in Canada, we haven't had one since the middle of January in the entire country.
But with these reassortments, I mean, with over 100 of them, does it seem logical to you that these would be natural?
Yes, I think they're natural, and it demonstrates the high degree of evolution of the virus in the wild population so that the wild population is actually much less risky now to us. They've had essentially a pandemic because these are migrating birds, but the birds have responded, and they have natural immunity, and it's harder for the virus to propagate in those wild birds now.
So what is it then with CFIA digging their heels in on this and not wanting new testing? And like, I mean, I know that
this is the absurdity, is that you have a situation where we have no data apart from the ostrich farm of the survival of any bird population that's domestic, commercial, in Canada, because we kill them off within a matter of days when we first detect any of them.
And that's who we're getting calls from, is so many vets saying, wait, stop. We actually have a recovered flock. Can we not do more research on this recovered flock and find out how it recovered? Can we not do some research? This is our first recovered flock because we were given the time to see what would happen if they stayed alive. And so I think that we have such a great opportunity here at Universal Ostrich to work with people like Dr. Pelech and other scientists on board that want to make change.
For any reasonable scientist out there, they would absolutely want to know why the flock recovered. Am I correct, Dr. Pelech?
Well, I think it's obvious. Even the Canadian Food Inspection Agency, under cross-examination, their experts all agreed that the birds have natural immunity.
Yeah.
At this point, if they survive past two weeks or 3 weeks without any further sickness, then it's basically it's been eradicated in the population. Now, on the one hand, they said that. On the other hand, they said, well, but these birds could still be shedding the virus.
Yeah.
So, I mean, the way natural immunity works is it takes out the virus completely. Otherwise, the animal will stay sick. So, but if the animal is recovered, then natural immunity is actually worked. So, in order to have shedding occur, where it's infectious, and this was the same with SARS-CoV-2 and COVID-19, sick people are the people that have the higher viral loads, and they're the ones that are going to be more able to transmit the virus. But if you have healthy people, their viral loads that they're transmitting is much, much less. So they're much, much less able for the virus to actually propagate in a new host.
And it's our natural immunity that is under attack. The threat is not by virus, but it's by the eradication of science. And we're really we're gonna run into a real problem if we keep making the mistakes that we're making on a global scale, because we're losing natural immunity. And that's what we're really fighting for here too, is that natural immunity holds a place. And we shouldn't be, this seems silly that we're fighting for it.
Well, we're not recognizing it. That's the problem. That's the problem we had with COVID-19. We didn't recognize natural immunity. No. We thought it had to come from a vaccine. When you look at the situation, we had about 14.6 million chickens killed in Canada in the last few years, over 160 million in the United States. That's why they're re-examining whether the culling, strategy is actually working. If you look at the number of outbreaks that we've had in Canada, it's interesting. The last 3 years, it's actually been seasonal, and the amplitude in terms of the number of cases hasn't really changed that much. So it's kind of like, the seasonal flu we see in people, we're seeing it in the birds, and it's probably in the wild birds. Now, these wild birds, they're migrating both heading south and heading north. So they're twice a year they're moving. Potentially they could be infecting basically the domestic animals if they encounter them.
Should be everywhere by now.
We're not seeing that. We're seeing only in the late fall is when we're actually seeing it in the outbreaks. And that's why, since January the 14th, there's no other outbreaks in Canada of H5N1 in a commercial farm.
And so we're not affecting trading. That's another thing.
I'm sorry. Go ahead, Katie. Say that again, please.
Or I was just going to say with that, we're not affecting trading. That's a big one that's being thrown on our plate, that we're affecting trading. And so, I mean, but due to that, because we're supposed to be 28 days, we're going for a 28-day avian influenza free status for Canada. We have not.
Again, that's ridiculous too, because the World Organization for Animal Health, they've set the guidelines that is supposed to be basically, informing the policy for the Canadian Food Inspection Agency. And what happens is, contrary to what's been suggested, the guidelines from the World Organization for Animal Health is that to have disease-free status in a zone, so that's another distinction, but in a zone, you have to be, if you did the calling practice, and it used to be 3 months, but as the threat got greater, believe it or not, they actually made the period shorter to 28 days. And so if you don't have any outbreaks in 28 days, then you get disease-free status for your zone. Now, if you don't do anything, if you let the infection burn out on its own, you have to wait a full year. But in many countries, that's what they do. They wait a year, and then they can regain that status for the zone. Now, the United Ostrich Farm is in its own zone. So the outbreak at the farm does not affect any of the other zones in British Columbia or Canada with respect to trade.
Right. That's interesting. But you are making glow-in-the-dark masks, though, aren't you?
Oh, man, are they fun.
Well, I think what was very interesting, the whole thing on the masks, I mean, my biggest beef was that the SARS-CoV-2 virus, it's about the same size as the influenza virus, about 0.15 microns roughly. The average pore size on, let's say, an N95 mask, for those ones, it's around on average 30 microns or bigger. So it's kind of like using a chain length fence to keep out mosquitoes. So the masks just don't work. However, they do have effects where you reduce, because of airflow, you reduce your oxygen coming into your body, and you're retaining more of the CO2 that you're expelling from your body. So you can actually raise the CO2 levels in your blood and reduce the oxygen levels in your blood. And your brain needs a lot of oxygen. But if you put the antibodies, if you coat the masks, then they can capture the virus as it's coming through. So you could actually make masks effective this way. And if you were in an environment where you're exposed to potentially the virus and you're wearing these masks, then you could then go later and you spray the mask with a reagent. And when you do that, then you can get the fluorescence to see that person has been in an environment where that virus is present.
So you could actually see it on the mask.
You can see it on the mask.
Okay, that's interesting. But you guys were not involved in that, so
No,
I don't even know why we're talking about that.
But I know how it would work. And so that is an interesting application and would work well with antibodies that are very stable to heat and to various conditions. So the ostrich antibodies would have been ideal for that purpose.
What else? I know that I think Dave, or maybe your mom, Katie, was telling me that there's so much more they're able to work on with the ostrich eggs. It's not just for virus. It could be for skin cream, let's say, or maybe even cancer treatments. I know there are other treatments. Do you know some offhand?
Sorry, what was the last part? I have somebody coming up the stairs.
Well,
Yeah.
We're talking about the other applications of the antibodies, and again, some of the fats that are in there. Ostrich eggs have a lot of other proteins that can be actually very useful, even as supplements. But one of the interesting applications I remember reading in the business plan for Studio is that you can have digestive enzymes that are actually being blocked by antibodies, so that you don't have as much fat, for example, being transferred into your body, so you can lower your cholesterol levels, for example.
So weight loss, could it be a weight loss supplement? Maybe
it could, yes.
Wow.
Because what it'll do is it'll block the ability of those digestive enzymes to break down the food product, so it can be absorbed into the gut, and so you would actually reduce particular fats, maybe some proteins this way. If that was, a situation where a person had, elevated levels of certain triglycerides, for example, and you want to bring that down, you could do it in this way.
And they've actually done studies. There was studies done with Dr. Tsukamoto on it with very successful results. So, and again, natural enzymes, using the antibodies and using, which they were actually talking about it really replacing Ozempic. It was that good without all those negative side effects that are now people are feeling for. Yeah, but
the Liberal government seems to want to get rid of all natural supplements and natural foods in this country, and the CFIA seems intent on stamping out healthy birds without testing. What conclusion can we draw from that, Dr. Pelech?
Well, I do believe that the people working at the Canadian Food Inspection Agency, they're trying to protect Canadians. They're doing what they think is actually is a strategy that they've received advice from other so-called experts on this. But when you start to think this through and you realize how, like with deeper knowledge of the mechanisms of how the viruses work and how the immune system work, and the data that's out there that you can actually study, the epidemiology data, you recognize that the strategy that has been adopted, it isn't really working.
No.
It's a huge waste. It's devastating small farms that can't afford what's being for example, what's proposed here is the Universal Ostrich Farm. They're being chastised because these birds are outside, running out on the land where they're encountering other birds that fly in. And this is a biosecurity threat.
Scary thought. That's a really scary thought, because again, in the case management conference, they mention all outside animals pose a risk because they're exposed to migratory birds. And so you can see where are we going with that?
Well, they wanted you to net the entire farm.
Right.
Like that would be ridiculous.
So the thing is that if you think about this, we already have animals that are in commercial sites, in buildings that are contained. This is very inhumane treatment of a lot of these animals, even if they're so-called free range. They're free range in a building, jumping on top of each other. They're not out there in fresh air, in green spaces, where there's an opportunity to be more in a natural setting for the animals. So their immune systems are actually more compromised when you're in a situation where animals are stressed.
Yeah.
It makes it even easier for infection to occur.
I was going to say those animals die faster. Those animals are dying faster. There's 40,000 chickens dead overnight.
Yeah. So this is it. I think if we were to adopt this concern about high security, biosecurity, don't forget that we've had farms for thousands of years where animals are roaming around on the land, and we haven't been as concerned. I mean, there are infections that go around historically, but the animals recover. And
Stephen, have you had I'm sorry to interrupt you. Have you had the opportunity outside of a courtroom to speak to any of these people with Canadian Food Inspection? And like from a professional standpoint, from your experience and everything that you know about immunology, like just to sit down and have a conversation with these people and try and reason with them in some way.
Right. And so what's happened is a number of us that are professors at the University of Guelph, which is, a university that has a veterinary school. I've actually been working with them as well, and they've been concerned about what's been going on at the ostrich farm. And so we've offered to actually try to work with the Canadian Food Inspection Agency to, look at the data. There's been no interest whatsoever in communicating with any of us to try to come up with a solution for the problem where we're offering our services. So, for example, I developed a methodology that we can track which parts of the various influenza proteins is the immune response being generated against. And that kind of technology, I'm happy to share and work with the Canadian Food Inspection Agency or any other, scientists that are interested in the immune response to influenza. But no, I haven't heard anything from any of my colleagues.
Do they have any scientists working at CFIA? Are there any actual?
Well, the irony of the situation is that some of the expert witnesses that were actually working at the CFIA were actually trained by some of the people that I'm working with.
In our scientific group that have been helping us for months.
They're actually pretty junior scientists that were the expert witnesses that
so you walk up to those guys and say, I should have flunked your ass when I had the chance. Well,
they didn't, not quite. Their background has very minimal immunology training.
Wow.
And so it's they may be very good vets, and there's a lot of expertise required to be a good vet. But being a good vet is just like a medical doctor. You can practice your craft, you learn how to diagnose and treat people, but the people that actually develop the new diagnostics and the new treatments are the research scientists. And they don't seem to have many research scientists.
Maybe that's why they didn't cross-examine any of our witnesses.
That's right. Yeah,
they wouldn't dare.
Yeah, but nobody wanted to ask me any questions.
Are there any misnomers, fallacies, disinformation left that we haven't gotten to tonight? Because I know that people, one of the reasons anyway that people were specially tuning in for tonight's show is because they wanted to hear you guys say, no, that's wrong, or that's not what's going on here. And I think we've pretty much covered everything that I knew of. But is there something that we may have missed that you guys know of, like open air biolab, or you're secretly running a fentanyl lab?
Yeah, I've heard that one too.
I would say the farm, people are talking about the farm being, these are lab animals. There's no lab at the farm. It's a farm. So for example, when I make antibodies against cancer proteins for research purposes, basically I produce the antigens in my lab in Vancouver, and then I send them to the United States to a farm where they have rabbits. They immunize the rabbits, and then they later send me blood from these rabbits, and then I purify out the antibodies from that blood. So you need a lab that actually does the purification to take it to the next step. The United Ostrich Farm is a place like that one in San Diego, which has rabbits, which actually will be taking antigens, putting them in the birds, and then providing eggs or egg yolk that contains the antibody, and then another lab or company has to then process it. So the idea that there's no viruses have been injected in the birds. They have been infected with the H5N1 virus. That's clear because of the sequence information, and the antibodies that are in these ostriches are extremely valuable at this stage, because this flock has survived being infected. They have had to have an antibody response to do it. And so those antibodies in these eggs can now be purified to actually facilitate biomedical research into the influenza virus, to create diagnostics, and potentially create therapeutics. And if we call all these birds, then this is a very valuable resource that we will have just squandered. And there's no reason for that at this stage.
So we're talking here about actual real scientists, done by actual real scientists here, taking as much time as they need to perfect something and try again if something goes wrong. It's not rushed. It's not manufactured haphazardly. All of the safety protocols are followed. Has anybody been approached by any of the big pharma giants like Moderna or Pfizer at all? And we want your eggs, and here's what we'll pay for them. Is there anything like that?
Well, I think that once we get more data on these ostriches, that will be recognized by the industry as potentially a very important source of what could be actually antibodies against the H5N1 virus.
I don't think they want that to happen, Dr. Pelech. I really don't. I think they've got other plans. That's only my personal opinion.
Yeah, no,
and maybe that's why the big stamp out, right?
Yeah, no, I think that there's actually a commercial opportunity here, because if in fact there's a concern of a farm being infected with the birds, if you have rapid antigen tests, and to develop rapid antigen tests, you need antibodies. In fact, you could determine, well, there's two types of tests, right? I mean, you have the PCR test, that's genetic. That'll only give you a positive result if you have an actual infection. The rapid antigen test will also give you a result, but you're testing for the protein, not the genomic material from the bird. But with that, those kind of tests, the rapid antigen tests, can be done anywhere. You don't have to have a special lab to do it, as you do with a PCR test. Now, the other test, the serological test, is for antibodies. That tells you that your birds have been infected and they have immunity against future infections. So both bits of information are useful, but these diagnostics are quite feasible using the materials from actually these ostrich eggs. And bear in mind that an egg is actually a sterile product. You know, if the egg was infected, then in fact the virus would propagate very quickly and the egg would go bad. So if you have a healthy egg, there's no virus in there, there's no bacteria in there, it's actually sterile. So it's a very safe way of actually producing the antibody product, even if you had the virus in the environment at one stage. It won't be in the eggs.
I'm sorry, I'm just looking at some of these comments and
Me
what I
too.
said. I mean, I can't even believe, like, it's mRNA. I don't believe any of this.
Yeah.
People.
Well, the virus, the influenza virus, is an RNA virus. That much is true. It's unlike some of the other types of viruses that use a DNA as genetic material. The RNA viruses, which includes RSV and the SARS-CoV-2, influenza is also an RNA virus. It's a little bit different. It's what we call a negative strand RNA virus compared to a positive strand RNA virus, which was what SARS-CoV-2 was. But these are small differences. But
it's not artificial like the mRNA. I mean, and there's none of that going on.
A positive strand virus is like an mRNA virus, but the influenza virus is a negative strand RNA virus. So that genetic material is not mRNA.
I think you guys have answered all the questions for tonight that we need answered. And judging from the comments that I'm looking at over here, even with Dr. Pelech telling us all of the things that he talked about tonight and Katie talking about, there's still people who are just never, ever going to understand or buy in because they are absolutely rigid in their belief that you're evil, Katie, and that's all there is to it. And
well, there's a lot of things to worry about in the world and to be scared of. And it's much more important that we focus our energies in directions where there is a real threat and we can do something about it. In the case of the H5N1, this is the least concern for me in terms of what's affecting our population and causing, sickness and death. There's a lot of other things that are happening right now that we should be much more focused on.
Yeah, I tend to agree with you. Thank you, Dr. Pelech, for joining tonight. And I want to get you on to talk about the books you've written and so independent of the ostrich farm conversation, I would love to hear your experience in life these past 3 or 4 years, because I've had other doctors on who have spoken about it, and it's been rough for them.
Yeah, no, I've been pretty lucky. I'm a full tenured professor at UBC, and I've published about 280 scientific papers in peer-reviewed journals. So it's kind of a bit hard to question whether I'm, competent on the subject.
You're Canada's answer to Peter McCullough, maybe.
Yeah, maybe.
Thank you for coming on today. Really appreciate you taking the time.
Thank you.
Okay. Katie.
Have a good night.
Before you go, Katie, I want to ask you, what's it like at the farm? You got a good mood there? Has there been any movement from CFIA, anything in the legal front, or is it still the same?
We just got a copy of our submissions that were submitted on Friday. We're really proud and happy with Omar's work, our lawyer, and we're just waiting. So it's a waiting game now here at the farm. It's another hot day. The sun is beating down in the field. The ostriches are happy, healthy, and loving it. And spending more time than we would probably ever want to try to. There's so much division. There's just so much division. Not at the farm, but just, on socials. And it would just be so nice to see everybody come together and save the animals. And nobody wants to see all the animals murdered or slaughtered. And I think it boils down to that. But I've learned a lot through this. We had the Atlantic is here today.
Oh, right.
Yeah, doing their article. And so he's just wonderful and learning a lot through that as well. But the whole mood on the farm is fantastic. Everybody's happy and just happy to be here supporting a good cause that we all believe in. So.
Okay. Please tell everybody I say hello.
Absolutely. Yeah.
Okay.
Thanks again. Okay.
All right. Thank you.
Okay.
All right.
Bye.
We'll talk to you. Bye-bye. So we do have a little bit more to cover tonight before we move to the extension, which is going to cover this. Of course, you know that there is some conflict over in the Middle East. When is there not? But it sounds like Israel, and it has been bombing Iran for quite some time, but it sounds now like the United States might get in on the action there. Donald Trump has said as much. He didn't say they will, absolutely, but he says, yeah, we're thinking about it. This is one of the reasons he left the G7 in Kananaskis a little bit early, and he's losing support from many of the people who supported him becoming president just a few months ago because he's coming off like a neocon. So we're going to cover why this is all going on in the extension tonight, which is coming up in about 15 minutes. I do have a couple of things to cover for you guys before we get there. Here's Chris Sky talking about the Iran. He just dropped this video about an hour and a half ago, talking about the Iran-Israel conflict. Interesting take, I think.
All of you that think Israel is so big and so powerful and blah, 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 blah. Yeah, they got the air superiority because they got the best stuff from the United States, so they can pretty much bomb anyone and get away with it, losing very few planes. Meanwhile, if they actually go to war with a country like Iran, and Iran wanted to actually invade Israel, what the hell could Israel do to stop them? Iran has an army of over 600,000 active duty troops. Israel is like 200,000, okay, to start with. Israel has like 400 tanks and 500 artillery guns. Iran has like 7,000 tanks and almost 10,000 artillery guns. Or sorry, opposite, 10,000 tanks and almost 7,000 artillery guns. So they could literally just march on Israel and literally walk over Israel in a matter of days or weeks. And Israel is sitting there all badass. And you know why Israel is sitting there all badass? Because they know the United States will fight and die for them. And all the European and everyone else that's owned and bought and paid for and blackmailed by Israel knows that Israel has nukes pointing at them. So if anybody does come to invade and all these countries don't go to war for Israel, Israel's going to kill millions and millions of people all around the world. And we're supposed to pretend that these are the people that are just defending themselves and these are the people that are okay and we should be backing them? No. And if Israel goes to war with Iran and the United States stays out of it, Israel lasts a couple of days, okay? When Iran gets to their doorstep, what are they going to do? They're going to nuke everybody because they know they're going down. That's what Israel is. They're a parasite on the world. They're a terrorist nation on the world. And I'm one of the only few people that's not afraid to say it.
Well, Chris, I think there's more and more people starting to wake up to that. And it's not necessarily the people. As a matter of fact, take out the word necessarily from that statement. You can say it's not the people, it's the leadership. And this has been going on for decades. Decades. It's time all of that nonsense stops. And the United States can stop it. So why is Trump suggesting that the United States will get involved on behalf of Israel in the Middle East again when he said that he wouldn't before? Well, we'll find out in the extension tonight for All Access Nighthawks at shadowdavis.com. In the meantime, what is Bill C-5? Jamie Sarconic, who wrote an editorial for the National Post, says, Mark Carney is demanding power to suspend all federal laws. What will he use it for? Federal fast-tracking bill would allow cabinet to exempt favored companies from any federal law, opening a massive door to corruption. And apparently the conservatives are in support of this. Liberals to pass major projects bill this week with conservative support. Chrystia Freeland thanks conservative Senator Leo Housakis for recognizing and highlighting that conservatives are supporting this legislation. In the meantime, liberal MPs are against this legislation. Here's Nate Erskine-Smith talking about it in the House of Commons today.
First, we should welcome greater parliamentary and civil society scrutiny. The government's proposed guillotine motion seeks to limit parliamentary debate at every stage of the bill. More concerning, it will jam all expert and public testimony and all committee scrutiny in less than 2 days. For what? For what? Consider that Parliament isn't currently scheduled to sit between June 20th and September 15th. We are rushing legislation through Parliament under the auspices of an urgent threat, but we aren't willing to put Parliament to work for what, an additional week to get things right? The debate on amendments does not need to be rushed. We could easily extend committee hearings by an additional week, provide resources for the committee to sit every day, and engage in a more thoughtful process to hear from experts, improve the bill, and pass it through the House by Canada Day.
Well, who's clapping? Who's actually clapping in the House of Commons if the Conservatives and most of the Liberals are in favor of passing this nightmare tyranny bill before they rise for the summer? And who's going to stop it from happening? Well, if the Conservatives are in favor of it, then all hope is lost, right? No. From APTN, First Nations threaten long, hot summer if Ottawa passes this national project bill. Again, Bill C-5 will desecrate burial sites. They can do, the Liberals, whatever they want. They can actually cancel laws. They can cancel laws that are on the books right now. If it has anything to do with development at all, and they see that it's getting in the way of a project they want done, then they can just say, no, the law doesn't exist anymore. Or they can also create bills and make them laws out of thin air without parliamentary oversight. That's what this Bill C-5 is. And the Conservatives do not seem to be opposing it at all. It gives Carney incredible powers. Why are they not opposing this bill? Is it possibly because they want that power for themselves when they finally take power? This will unhinge our democracy. It has the capabilities to do that anyhow. And the Conservatives are just letting it slide. So the Indigenous say no. A First Nation leader from Northern Ontario had a warning for the federal government today at a protest over Bill C-5, the federal government's proposed national project building legislation. If you pass this Bill C-5, it will be a long, hot summer, said Alvin Fiddler, Grand Chief of the Nishnawbe Aski Nation, an organization that represents 49 First Nations in Northern Ontario. We will not sit idly by and watch any government, whether it's Ontario or Canada, to come to our territory and take whatever they want because it's ours. Fiddler is also in a fight with the Ontario government over the province's own legislation that gives it sweeping power to override environmental and municipal laws if it deems a project to be important. On Parliament Hill, about 350 people brought signs and ceremonial drums to help make their point. Isn't that something? I can't even believe that more people are not concerned about this bill, and only 350 Indigenous people showed up at Parliament Hill to protest. What's happened to our country that we can't stand up for ourselves anymore? It's absolutely outrageous. Mark Carney is giving Ukraine another $2 billion in aid. This was announced kind of slyly because we all know that Trump has been working on some kind of peace deal with Ukraine and Putin over there in Russia. And so Trump left the G7 early this morning or late last night because he had other things to do, like, bomb Iran maybe. And so when Trump leaves, Zelensky slides in. And Carney says this.
I would like to start by condemning in the strongest terms the latest outrage, barbarism from Russia, and express my deep condolences and those of the Canadian people to the families in Kyiv who were affected. This underscores the importance of standing in total solidarity with Ukraine, with the Ukrainian people. And we had a discussion last night amongst G7 leaders. The President and I will discuss more, and then of course we'll have the direct discussion in a moment together. But we underscored the importance of using maximum pressure against Russia, who has refused to come to the table. The President has led unconditionally to come to the table in search of peace. And maximum pressure includes financial pressure, and Canada is announcing today several measures, sanctions, first and foremost, against a number of individuals in Russia, against over 40 entities in Russia and beyond that are trying to contribute to the evasion of sanctions, the sanctioning of over 200 vessels in the shadow fleet that Russia is using, again, to try to evade these sanctions. That's the first thing. The second is we are providing additional military assistance.
Where are we going to get $2 billion? Oh, he says it's going to come from the fund of all of the money that we've seized from Russia that I'm sure has already been spent or may not even exist in the first place. Give it up, man. Give it up. Just stop it. This is not our fight. Stop it. But if Pierre Poilievre was up there, if he was prime minister right now, he'd be doing the same thing because he believes in that stuff. And I don't understand why every politician in this country is captured by that ideology. I like Danielle Smith. I do. I think she's probably the most courageous politician we have in this country. But look at this. It was an honor to welcome President Volodymyr Zelensky to Canada for the G7. Ukraine's not a G7 country. Here in Alberta, our historical connection to Ukraine runs deep. Its culture, its strength, it's woven into our fabric of our province. Alberta stands with Ukraine and remains committed to supporting humanitarian aid. Could that be a political move because there are so many voters in Alberta of Ukrainian heritage? That's why they call Edmonton Edmundchuk. But then again, Edmonton is a NDP stronghold. What are you doing, Danielle? Just say it like it is. We can't afford this nonsense any longer. A couple more things before we move to the extension tonight. Remember the video I showed you last night of that protester in Los Angeles being run over by that frustrated motorist? This is during the big No King protests in Los Angeles, the big anti-ICE protests all over the country in large cities down there in the United States. Well, in Florida, they have a different look on protesters swarming cars. Here is Governor Ron DeSantis.
Have a policy that if you're driving on one of those streets and a mob comes and surrounds your vehicle and threatens you, you have a right to flee for your safety. And so if you drive off and you hit one of these people, that's their fault for impinging on you. You don't have to sit there and just be a sitting duck and let the mob grab you out of your car and drag you through the streets. You have a right to defend yourself in Florida.
Excellent. Is there a way to get to Florida without being an illegal? Inquiring minds want to know. I'm going to throw this up here. This is an interesting story. Interesting that CTV, left-wing corporate, covered this as well. Like cutting their own throats, maybe. U.S. news consumers, and this is happening in Canada too, to a lesser degree and at a slower pace, but it is happening. I'm happy to tell you. U.S. news consumers are turning to podcaster Joe Rogan and away from traditional news sources, according to a report. Prominent podcasters like Joe Rogan playing a bigger role in news dissemination in the United States, as are AI chatbots contributing to the further erosion of traditional media, according to a report released on Tuesday. In the week following the January 2025 U.S. election, more Americans said they got their news from, inauguration. The election was in November. More Americans said they got their news from social and video networks than from TV and news websites and apps, the first time that shift has occurred, said the report. Traditional U.S. news media increasingly risks being eclipsed by online personalities and creators. The Reuters Institute for the Study of Journalism said in its annual digital news report, which is based on an online survey of almost 100,000 people in 48 markets, including the United States. The trend is particularly acute among young Americans. Over half of the people under age 35 in the U.S. are relying on social media and video networks as their main source for news, the report found. Across the countries that the report surveyed, 44% of people aged 18 to 24 said these networks are their main source of news. I wonder what kind of news they are consuming. How to take the perfect selfie. 1/5 of a sampled group of Americans came across news or commentary from Joe Rogan in the week following the presidential inauguration. Well, 14% of respondents said they had come across Tucker Carlson discussing or commenting on news during that period as well. Carlson now generates blah, 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 blah. Also, Megyn Kelly, Candace Owens, Ben Shapiro on the political right, Brian Tyler Cohen, and David Pakman. Oh, that guy. If you want your blood to boil inside of 15 seconds, watch a video that guy's made. David Pakman and Brian Tyler Cohen's another one. The vast majority of the most followed commenters who discuss politics are men, the report found. Again, happening in Canada, just not at the rate that we would like it to happen. But it is happening. Now, you guys remember John Julian? He's the entertainer from Vancouver who used to do all the Justin Trudeau impersonations. Well, now that Justin Trudeau is gone, John has switched, or maybe just worked on some new impersonations, and he's doing a pretty wicked Jordan Peterson these days. Watch this.
Cleaning ovens, the enablers of young, lazy bachelors who cloak themselves in the guise of moral virtue. The same self-aggranding thugs who leave greasy piles of dishes with hedonistic baked-on cheese and egg, which is not insignificant. And although their rampant intake of soluble fiber assures their untrammeled regularity, they are often betrayed by their skid marks. So take the damn plunger. The toilet is like the mind, occasionally clogged and needing a good flush, and should glisten in the best possible light, particularly after being replete with the horrors of men whose urinary aim is worse than a totalitarian hellhole leader's water cannon.
Not bad. Not bad. I don't think he squealed high enough, but maybe that's only one part of it, and I missed that part. The extension. Why is Donald Trump threatening military action against Iran right now? At the urging of Mossad, the urging of Israel. He didn't want to do it before, but now suddenly he does. So why? We're going to look at the possibilities in the extension tonight on shadowdavis.com. So for you Nighthawks, I will see you there in about 30 seconds, maybe closer to 2 minutes. For the rest of you, thank you for joining the show tonight. I will catch you tomorrow night at 8 o'clock Central for another Shadow at Night live stream. Bye for now.